Morning everyone and welcome to my YouTube channel. So here today we are going to be looking at our nucleus, our Golgi body, and our rough and smooth endoplasmic reticulum. So the nucleus, we know that the function of the nucleus is to control all the cell's activities, and also it stores genetic information. So if we look at our nucleus, it is membrane bound, meaning to say it has got its own membrane. So the membrane that the nucleus has is the nuclear envelope. The nuclear envelope has got pores, the nuclear pores, which are more like spaces in between the parts of the envelope. Now, the function of the nuclear pore is to control what leaves and enters the cell. So now when we look at our structure, we have other parts of the nucleus which we are going to call the nucleosome. The nucleosome is the liquid part of the nucleus. So in the nucleosome, this is where things such as our DNA is suspended. So the DNA will be suspended in here, like that. Then also other structures such as the dense part of the nucleus, which we call the nucleolus. So if you look at the chromatin here, the chromatin is more like the one that stores the DNA. It's more like the DNA when it wraps around histones, it is able to form the structure that we are going to be calling the chromatin, like that. Then when we also look at the other structure, which is our nucleolus, which is the dense part of the nucleus, this structure is responsible for the synthesis of RNA. This type of RNA is the RNA which we are going to be calling the ribosomal RNA. So this ribosomal RNA is the RNA that is now transferred to what we are going to be calling our rough endoplasmic reticulum. So this is the basic structure of our nucleus. Our review: nuclear envelope has got spaces in between which we are going to be calling the nuclear pore, which allows certain substances to enter and leave, such as the RNA. It can be moved out and all that. We have also talked about the nucleoplasm. This is the liquid part of the nucleus, in which substances such as the chromatin is suspended, and other things such as the nucleolus, which is the dense part, and it is responsible for the formation of ribosomal RNA, which is able to bond afterwards to the rough endoplasmic reticulum. So our 3D diagram would look like this. The liquid part, which is our nucleoplasm, the nuclear envelope, then we have the ribosomes, like that. Now from there we are going to be looking at our rough endoplasmic reticulum. So very close are the endoplasmic reticulum. So depending whether the ribosomes are there or not will determine whether they are the smooth or the rough. So the endoplasmic reticulum would look like a structure like this. So this is our smooth. When we attach our ribosomes like that, then it will be called the rough endoplasmic reticulum, like that. So that is the general structure of our endoplasmic reticulum. So it might look like this in 3. We might have such a situation. So these are the ribosomes. So if the ribosomes are there, then we have this type of the structure. Then we are going to call this the rough endoplasmic reticulum, because they have the ribosomes. So our rough endoplasmic reticulum, since they have the ribosomes, they are responsible for making, or rather the word that we are going to be using is synthesis of proteins. So they are responsible for the synthesis of proteins. When these are made, they are now sent to the Golgi body, which is the Golgi apparatus or the Golgi body, in other words. So now we also have the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is responsible for lipid synthesis. So lipid synthesis. Now when these are made, they are now taken to the ribosomes via what we are going to be calling transport vesicles. So the vesicles will be able to more like carry the lipids and the proteins to be taken to the Golgi body for modification. So when substances are made by the smooth or the rough, they are transported via the transport vesicles. So our vesicle like that, which is this one, then it is moved to the Golgi body. When it enters there, whatever is transported there, it is going to undergo number one, modification and packaging and sorting. So now when we look at our modification, so under modification we are going to be looking at how it adds some sugars such as glucose and other substances to produce a glycoprotein. So remember, let's say the proteins come to the Golgi body. The Golgi body will add certain carbohydrates to form what we are going to call a glycoprotein. The glycoprotein, which is used by the plant cell or by the cell in signaling and also identification. Also, if the lipids are taken to the Golgi body, they can also undergo modification by the addition of carbohydrates to also form a glycolipid. So glycolipid like that. Then when we look at the receiving part of the Golgi body is called the cis phase. Then the end part is called the trans phase. So other substances are sent away from the Golgi body, such as those substances that are harmful to the, let's say, the cell. They are taken outside the cell. Sometimes it's not just those that are harmful. Some of them are just taken towards the cell surface to make, let's say, the cell membrane and among other things. So the other function of the Golgi body is to help in sorting. So under sorting, it will be able to more like direct the proteins that are made to either the cell surface which we are calling the cell membrane, and also sometimes the cell surface is just more like the surface of the membrane, and sometimes it might be the plasma membrane itself, which means to say in between the plasma membrane, some of the proteins might be directed there to form one or two things. Then the other thing that is very, very important is that the Golgi body produces what we are going to call the lysosomes, which we talked about in the previous video. So it helps in the formation of lysosomes by packing some of the enzymes in a vesicle. So that those are the functions of the Golgi body. Thank you very much for watching. Hope you would like, subscribe and share for more. See you in the next tutorial.